Well, hello everyone and welcome back to Blackboard Biology. This is our DNA series, lesson 2, specifically the structure of DNA. Now in our last video, we looked at some of the history behind the discovery that DNA was in fact the genetic material that made up genes that controlled traits in all of our cells and in basically every form of life on Earth. They didn't quite realize that fully at the time, but that's true. And so what we're going to do is in this video is look specifically at nucleic acid structure. So nucleic acids are DNA and RNA. They're found in the nucleus of cells, and RNA is found also outside of the nucleus, but it is produced in the nucleus, and they're slightly acidic, so hence nucleic acids. And what you need to understand right away is that they are long chain molecules. DNA and RNA are what are known as polymers. Now, a polymer is just that, a long chain molecule, and it is made up of many connected monomers. So if you think about a chain and the links in a chain, that is what the monomers are. So I like to think of them as building blocks, as links in a chain, as like little Lego pieces. I can make all kinds of Lego structures with little pieces that I put together into larger structures. So a monomer is a smaller molecule that when you link them together make up larger polymers. Specifically for DNA and RNA, these monomers or building blocks are known as nucleotides. Now you may be thinking, okay, I've never heard the word nucleotide. What in the world does it look like? Well, we can kind of visualize it like this. Specifically, this is a nucleotide that you could find in DNA. Now, here's how I would know this based on this right here. So as you can see, there are 3 basic parts to it, and this first part that I want to draw your attention to right here is a 5-carbon sugar. Here's how I know that it is DNA, because specifically this sugar is deoxyribose. Deoxyribose, as in deoxyribonucleic acid, or DNA. So in this case, I'm saying this is deoxyribose, and if it were RNA instead of deoxyribose, it would just be plain ribose. The only difference is deoxyribose is missing an oxygen atom, hence deoxy. So that's the first part of a nucleotide. Now the second part that I want to draw your attention to is what is known as a phosphate group. All right, and so this right over here, we've kind of represented with a little circle here. This is a phosphate group. It is a polyatomic ion made up of phosphorus with 4 oxygens attached, and it is covalently bonded to the 5-carbon sugar right here. And so it bonds with that, and together these two form the backbone of the DNA molecule. We'll see how that works in just a minute. Well, the last part that you'll see here in a nucleotide is very, very important. It is not so much structural as it is, this is the informational part. This is what is known as a nitrogenous base. And you'll notice that it has a letter on it. The letter in this case is an A. A for adenine. That is the nitrogenous base, adenine. And so what you'll find is that the nitrogenous bases are what make up the code. Now, if you remember from our last video, we talked a little bit, people were on the fence. They kind of had this big debate going, is it DNA or is it proteins that are the genetic material? Well, DNA, these bases are what form the code, the letters of the quote-unquote DNA alphabet. Now, our alphabet in English has 26 letters, and proteins have basically 20 different letters to work with, 20 amino acids that are found in life on Earth. But DNA only has 4. You have A, T, G, and C. Adenine, thymine, guanine, and cytosine. And so they were like, oh, there's only 4 letters. It can't produce as many combinations. Perhaps this is not it. But these 4 types of nucleotides, again, the nitrogenous base is what makes them different. These 4 types of nucleotides are what make up the genetic code of all life on Earth. And so I want to dive a little bit into some of the history of kind of discovering the specific structure and how all of this kind of came to be. So many people, there was a huge race to be the first to discover the structure of DNA. And before kind of this race fully set in, before they totally knew that DNA was the genetic material, there was a guy by the name of Erwin Chargaff who was working with DNA and studying these things. And he studied many different DNA samples from just all kinds of different organisms. And he found some very interesting things about these bases: adenine, thymine, guanine, and cytosine. And here's what he found out about this. A and T, adenine and thymine, the percentages of adenine and thymine in any given DNA sample were always just about equal. And the guanine and the cytosine were always just about equal. And so this became known as Chargaff's rule, that the adenine in a sample of DNA would always be equal to the amount of thymine, and the amount of guanine would always just about be equal to the amount of cytosine. Chargaff's rule. Now, he had no idea why, and nobody else did for many more years. But Chargaff's rule was very important in understanding the structure later on. So many years later, in the early 1950s, there was a young woman by the name of Rosalind Franklin. And she was working on DNA using a process known as X-ray crystallography. Now, I have to admit, I am no expert when it comes to X-ray crystallography, and trying to understand the results of X-ray crystallography is a little bit beyond my thing, but I've done some research and I'll try to help explain it the best I can. So what scientists do when they want to look at things and see very small structures, we often use microscopes, but you're limited by the wavelength of the light you're using to try to view it. And so when you want to look at a specific molecule and its structure, regular light just doesn't work. But X-rays do. They have a different wavelength, a different frequency, and so these X-rays can in fact strike very small objects. And I'm talking like specific repeating patterns of atoms and molecules in a crystal. And so what they would do is they would use a radioactive element that would give off X-ray radiation, pass that through a laser to kind of amplify it up and everything, and shoot an X-ray at a crystal structure. Now, why crystal? Well, because crystals form repeating patterns. They didn't really use a crystallized form of DNA. She just took a bunch of DNA strands, aligned them all together as best she could. It was about a human hair wide of several strands of DNA. And as those X-rays hit it, the X-rays would bounce off of things in different patterns and strike X-ray sensitive film and kind of produce this image. And then you would analyze the image. Well, Rosalind Franklin was doing this research at King's College, and she had an undergrad student that was working with her. Her supervisor was Maurice Wilkins. And in time, she got better and better. She was a meticulous researcher, meticulous, true scientist. And in time, she produced this image right here. This is what has become known as the infamous Photo 51. Now, first glance, this means nothing to me. Okay, I have to do research to try to understand what in the world it means, but to an expert, somebody who does X-ray crystallography, this speaks volumes as to the structure of DNA. This photo was instrumental in helping scientists conclude the structure of DNA. And so I found another photo where somebody kind of helped us understand a bit how to interpret this. Now, before we go to that, though, I want to say that Rosalind Franklin, she did this, and she spent some time examining Photo 51 and trying to understand it, and made some found some key insights in it, but she was right on the cusp. She stopped just short of really knowing for sure the total structure of DNA. And unfortunately, in 1952, she was going to be moving to a different location to continue work with X-ray crystallography. She didn't just work with DNA, she worked with lots of different molecules. She worked with viruses with X-ray crystallography. And unfortunately, 6 years later, in 1958, she died from cancer at a very young age, but she was about 37, I believe. She died from cancer, and so she never really got credit for most of this work that she did. But this photo her supervisor, Maurice Wilkins, as we'll find here in a moment, showed this photo to some other scientists who were working on the structure of DNA, and it was the final missing piece of their puzzle. And so here's why. Looking at this photo, if you understand X-ray crystallography, which I don't very well, but what I do know is that the helical structure of DNA, kind of this zigzag pattern that you can see, is formed because of the way that the helices align and you get these major and minor grooves. When the X-rays would strike this, they would come off at perpendicular angles to the helix. And so it produced this X kind of shaped pattern you see here on that. And so people who are familiar with these things, they would conclude pretty quickly from looking at this, okay, we are looking at a helical molecule here, and because of some other intricate details, it is not just one helix, it is a double helix, which has become synonymous with DNA. You hear DNA and people automatically think double helix, but this was what really clued them into that. You see these layer lines that it's talking about over here. These layer lines help them conclude many things about the fact that there was major and minor grooves. It says that there's a missing layer line here on the what would be the fourth layer, and that kind of showed them that, hey, there's some things that kind of interference here because of this minor groove, and so you don't get that there. Also, these really, really dark spots up here, again, I'm not entirely sure why, but that clued them in as to the fact that the nitrogenous bases were repeating and that they were in the middle of the molecule, and these layer lines helped them conclude that the phosphates were the backbone and they were on the outside. So they concluded it was a double helix and that the backbone of it was on the outside, while the bases, what they later discovered was the code, were on the inside. This was super, super instrumental because while Rosalind Franklin didn't quite draw the exact same full conclusions, because she was an experimentalist. She wanted to work with empirical data. She wanted to gather her own data and conclusions. She did not like working with scientific models very much. Well, there were a couple of fellows that you're probably familiar with, Watson and Crick. They were working on various models, working with scientific modeling to try to discover a working model, a working representation of the structure of DNA. And in 1952, shortly before Rosalind Franklin was to begin working somewhere else, her supervisor, Maurice Wilkins, showed Watson and Crick photo 51 and some of her other work. Now, there's debate as to whether or not he should have done that. He really wanted. He thought her work was. It was kind of a compliment to her work in one way because he thought it was so instrumental and so important that he wanted to show them. But in another way, he didn't get her consent to do so, and she hadn't published it anywhere. So it was kind of a big no-no in scientific circles. But anywho, they saw it, immediately recognized, hey, this is a double helical structure. The phosphates are on the outside. They went back to work with their previous research and data and their models, and they quickly were able to deduce an accurate structure of DNA, which was later verified by Maurice Wilkins and his colleagues at King's College. And in 1953, Watson and Crick published a journal article with a photo of their model. It was a very short article and kind of explaining the structure. And quickly after that, they followed up with more articles of how this structure explained the functions of DNA, how it was able to replicate itself, how it was able to hold its shape, and how it was able to store and transmit information. And so specifically from these models, how could we kind of visualize and how could we kind of see these things? Now, I do want to say one more thing actually before I look at the structure. In 1962, these three guys, Watson, Crick, and Wilkins, received the Nobel Prize in physiology or medicine for their work on DNA and discovering its structure. Franklin had died in 1958. She could not share in this prize because it is only awarded to individuals who are still alive at the time that it is awarded. And so she never shared in that. She never got to win the Nobel Prize, even though they likely could not have figured it all out without her. So it's kind of a shame. But they discovered this structure right here. Here's a kind of a way to represent it. you will notice very quickly that we have up here what appears to be, there's two strands and they kind of run in opposite directions, and that's exactly what we observe. Up here, this phosphate right here attaches to what is known as the 5' carbon of this deoxyribose. Down here, this corner of the deoxyribose down here is what is known as the 3' carbon. And so DNA is said to run from 5' to 3'. Now, if you look at the strand on the right side, here is a 5' and here's the 3'. And so they actually run parallel but in opposite directions. So they are said to be anti-parallel strands. Anti-parallel strands. That is super important. That explains why DNA can replicate itself. It also shows, this explains Chargaff's rule, because what we notice here is that A and T always pair together. And I'm representing those bonds that connect them with dotted lines because they are weak bonds. They're called hydrogen bonds. They're not the same as these covalent bonds over here, which is good because we actually need to be able to, split the DNA down the middle at times to replicate and get the information out of it that we need. A and T always pair together, and they have 2 hydrogen bonds that connect them. G and C always connect together, but they have 3 hydrogen bonds that connect them. So this explains Chargaff's rule, and this is known as complementary base pairing. This explains so much of how DNA is able to function in the way that it does, how it's able to replicate, how it's able to pass along information, how it's able to lead to the forming of RNA and eventually proteins and things, as we'll see later in future videos. So I hope that this video has helped you understand the structure of DNA a bit better, how it's formed of nucleotides, how nucleotides have phosphates and sugars and nitrogenous bases, and a little bit of the history that led to these discoveries. As always, I want to invite you, like, subscribe, leave comments below. I'll get back to you. And as always, thanks for watching.